All right, it's my pleasure to introduce Dr. Cheyenne Smith. She's a postdoc researcher at the USGS Eastern Ecological Science Center at the Leetown Research Laboratory in Kearneysville, West Virginia, working with Dr. Vicki Blazer's lab group. She's been an active member of the fish health section since beginning her master's degree in 2015. And additionally, she holds a steering committee position for the Immunotoxicology Interest Group of CTAC. Outside of research, she spends her free time doing toddler things with her 2-year-old and practicing martial arts or training dogs. Awesome.
Thank you,
Take it
Nick.
away, Tran.
Thank you, Nick. So I'm going to be talking about the immunotoxicity portion of our wild fish health assessments. Why were we interested in the immunotox in the first place? Well, chronic health challenges to smallmouth bass have been occurring in the Potomac and the Susquehanna since 2002 and 2005, with high prevalences of visible lesions. Some you can see here, multiple bacterial pathogens, sometimes Flavobacterium, some Aeromonas species, heavy parasite loads like you see here in the spleen and in the gills over here. Opportunistic fungal infections, we see viruses, largemouth bass virus, sometimes adenovirus, which is related to the blotchy bass. And often we see co-infections of all of these, plus signs of oxidative stress. So here's some macrophage aggregates in the spleen tissue, reproductive endocrine disruption, which is testicular oocytes that show up in male fish, and also population declines. So for instance, from 2013 to 2019 at one of our sites, Antietam, you can see the decrease in the catch per unit effort. And so all of this together led us to look and wonder if immunosuppression was occurring. So 23 years ago, the fish were happy, the anglers were happy, everybody was good. And then in 2002, started noticing deaths and disease. So that's what led us to look at immunosuppression in context with the ecosystem and complex interactions of pathogens and chemicals that the fish may be facing, and look at whether or not this is related to an elevated disease incidence in smallmouth bass. So the study design, first, we chose immune endpoints and developed assays for smallmouth bass. Then we applied them at field sites. And now we are using those findings from the field to inform and guide our laboratory studies. So which chemicals should we look at, what concentrations, maybe what mixtures, and this will help us understand some of the underlying specific immunotoxic mechanisms that may be happening. So this is a great figure of kind of the overall methods that are occurring in the immunotox world. Encircled are the ones that we chose for our study. So you can see how they rank above on the transferability across species and the frequency of assessment continuums. We're looking at the lymphocyte proliferation, respiratory burst activity, phagocytic cell activity, in terms of bactericidal activity, and then also immune-related gene expression analysis. So for this immunotox study, we did two different studies, one from 2016 to 2018, where we had 4 sites where we kind of intensely monitored the fish health, but also the water quality assessments. And then in 2019 and to 2021, we sampled West Virginia sites. And you can see there's a range of land use among all of these, and also important to note that this one site in the middle here, SB3, was part of both of these studies. So at the end, I'll show you how we're kind of using that data from that long study from 2016 to 2001 and what we can kind of pull from having such a long study. So the study design includes a field component and a lab component. So for the field component, we get that ecological relevance of everything that's happening. We have the ability to do long-term monitoring, and then we can use the data from that for informing our laboratory studies. And the pros of having a laboratory study, of course, are having the controlled environment where we can control temperature, dissolved oxygen, and food availability. We can control the chemical concentrations and mixtures. And also it allows us to confirm chemical and pathogen history of the fish, which we cannot do in the field. So it's more of a way to develop a cause and effect relationship versus just a risk assessment in the field. So using these two together when you're trying to design an immunotox study is pretty valuable. Immune endpoints, the ones I'm going to talk about today are respiratory burst activity and lymphocyte mitogenesis. So these are both they have a unique ability to give us a background response. So respiratory burst activity, as you can see in this video, you have a phagocytic cell. So in this case, this is a neutrophil. It's digesting a bacteria right now, putting it in a vesicle. And then the respiratory burst is actually the production of chemicals called ROS, reactive oxygen species, that actually are what degrade that bacteria. And then the cell can spit out the dead bacteria. So this is a way for us to measure generalized oxidative stress in phagocytic cells. And this is an innate measure. So innate immunity is what you're born with. On the other hand, we have lymphocyte mitogenesis, which, as you can see, these are lymphocytes, so T and B cells. This is part of your adaptive immunity. So this is what you develop over the course of your life based on what you're exposed to. And fish are the same as humans. And it just measures the ability of the lymphocytes to proliferate or replicate in response to 3 different mitogens that we're using. So like I said, both of these assays I'm going to talk about because they have a unique ability to monitor the background response separate from the stimulated. The stimulated is calculated as a fold change over the control wells. So those control wells are what actually give us a lot of information because they're field cells, and so it gives us a baseline of where they're coming from. And so the immune response in general is a delicate balance. The background gives us a baseline, especially important for wild fish when we lack the knowledge of exposures or their history of disease. And so this background response may be as or even more important than the stimulated responses. So I'm going to talk about site comparisons of the background reactive oxygen species. So this is that top 2016 to 2018 study sites. And it's the Susquehanna River sites versus the Potomac River sites. These are both rivers in the Chesapeake Bay watershed. And the background ROS was higher at the Susquehanna sites, the one which are ash strip. But what does this mean and how do we interpret these findings? We really can't unless we look at it in context with what else is happening biologically, environmentally, and chemically at each site. So for instance, if we look at the Potomac sites, we look at the transcriptomics of the immune genes and found that 14 genes were actually downregulated at the Potomac sites compared to the Susquehanna, and these were related to different immunity things like inflammation, contaminant metabolism, microbial metabolism, the adaptive immune response, and the general stress response. Also at the Potomac sites, they experienced higher parasite burdens and co-infections and myxozoan parasites. So this is important because myxozoans compared to helminth parasites, which are larger like worms, trematodes, cestodes, the myxozoans are smaller. They can evade the immune response more than those larger helminth parasites. And so that could be part of the story and why the Potomac sites show a lower background, something to consider. Also, we have to consider this. So this site is the Susquehanna site called West Branch Mahantongo, and it has the highest background ROS. So if you look deeper into it, at the surface water chemicals, this is for 3 pesticides that are most commonly detected at all of these sites. And what we see at this site, West Branch Mahantongo, is that the surface water actually shows consistently moderately high levels of pesticides throughout the entire study from 2016 to 2018, which is not seen at these other 3 sites. So now, well, Antietam does have the highest levels, but it's only at the beginning of the study and doesn't continue on. So what are some potential consequences of having a high background reactive oxygen species or respiratory burst? One, lack of efficient bacterial killing. So at Antietam Creek over here, we had a negative correlation for both bacteria. So we test 2 bacteria, one that we have isolated from them, so they could be exposed to already, and one that we have not isolated from them, and it's actually a cold water species, so they are less likely they should not be exposed to it. There's a negative correlation indicating that as the ROS, those chemicals go up, they're actually seeing a decrease in the bactericidal activity. That indicates that there's actually too much ROS and there's not an efficient bacterial killing response like you see at the South Branch, where as the ROS goes up, so does the bactericidal killing. And that's what we would expect because the mechanism of these chemicals is to kill bacteria. Another potential consequence is could be oxidative stress and damage to the host cells, including the cell death. So this would happen when there's actually an imbalance. There's going to be too much of those ROS chemicals and not enough antioxidants to reverse the damage from those. So these two sites are sites from the Susquehanna sites. And the bactericidal killing was relatively low from 2016 to 2017, and no relationship with background ROS until we see 2018 at both of these sites. So therefore, another consequence at these sites may have been that the background ROS was actually too high for optimal killing, and maybe even the cells not functioning correctly at all until 2018. Just these are things to keep in mind when looking at immunotoxicity of wild fish. Now, if we look at the site comparison of the background mitogenesis, so this is that adaptive arm of the immune response. These are those same sites, and the lowest background is at Antietam Creek. Now, when we look at the transcriptomics for this, 19 genes, immune genes were downregulated and 3 were upregulated at this site. So this kind of leads me to think that Antietam, this site, may be a potential hotspot for us to continue monitoring long-term, not only because it had the lowest background immune responses, but also because it had the highest myxozoan densities and parasite co-infections. It has the highest percentage of surrounding agriculture and developed land use. And also it had the highest concentrations of pesticides detected in the surface waters. And even though it wasn't consistent, it still had the highest concentrations. So when we start to think of this all together, the first thing to look at is immune function correlations. So how does the immune function correlate with the other data that we have collected? The other data that we have collected, some examples, are the Health Assessment Index. So this is a number that is assigned to the fish based on all of these different tissue types, the fins and body surface, gills, eyes, liver, spleen, and the inside organs too, anterior kidney, posterior swim bladder. And they get assigned a number, and that equals the Health Assessment Index. So we have seen mixed correlations with the Health Assessment Index with seasonal differences, some sex-based differences. Same thing with land use. So these are two different immune parameters correlating with the different types of land use, but only for certain seasons. So forested only correlated with this immune test in the spring, whereas the other more developed and agricultural lands were only correlated in the fall. So I just want to point out that we are seeing seasonal and sex-based differences that have to be considered too. And if we look down here, this is total mercury. And so in addition to the water surface chemistry and the pesticides in the water, these fish are also being exposed to bioaccumulative chemicals such as mercury and PFAS. And here's an example of how one immune function test is correlating with the average total mercury in the liver and the muscle. And so these are just preliminary results because I only have 3 data points, but you can see there is a sex-based difference. There's really no correlation with the females, and there is a trend with the males, although it's not significant, but I think it's because of my data points. So we're seeing the seasonal and the sex-based differences. But then we have to remember, this is actually a really cool website that this guy, he does correlations with just random study things. So we have to remember that correlation does not equal causation. So for instance, the per capita consumption of margarine definitely does not cause the divorce rate in Maine to go down, but it looks like it does. So it's just to point out that there are other things we have to consider. And those other things, when you look at wild fish, are lot of things, such as parasites and pathogens, which we are doing the prevalences of, season and climate, also population densities of the fish, of the parasites, of pathogens, also of people, genetic diversity of the fish, and maybe their epigenetic responses, sex and age of the fish, because I was telling you we are seeing sex-based differences, and also age has been correlated with immune function too at some of the sites. Land use. Also, we have to consider the chemical exposure that we can measure both in the water and in the fish, and also start to look at early life exposures. And so all of that is different from a lab. It's different from a lab experiment because we have all of this to consider and put together like a puzzle. So just as an example of that South Branch site that I told you I was going to get back to for the long-term monitoring and how we can start to tease out, well, maybe early life exposures do play a role. So for instance, we have the sampling dates from 2015 to 2021. We have the concentrations of atrazine and metolachlor, which are two of the pesticides that are most commonly detected in the lines. And then this orange bars are the background ROS, the average for each sampling period. So each one of these bars is 20 fish that we sampled on a certain day from this site. And in the box is the average age of the fish from that sampling event. And so for instance, this 2019 fall sampling with an average age of about 4, we can go back and say, this high response, well, they were exposed when they were just born to high levels of pesticides. Similarly, this sampling, 3-year-old, can go back to lower incidences of pesticides. And again, with the following one, these are about 3 years old in 2021, we can go back and say, okay, well, maybe they were exposed to low concentrations. So this is just the beginning of considering all of this type of stuff. It could be coincidence, it could be related, but definitely have to consider it and try to look at it in the future when we plan these studies out. Because as we know, immune function, it can be affected, chemicals can affect a baby more than it can affect, or differently than it can affect an adult. Also, what we have discovered is that sometimes the pesticide metabolites can actually be detectable at significantly higher levels than the parent compounds. So for instance, atrazine right here, this metabolite was detected at nearly 100 times the normal concentration of atrazine, which is the dotted line. And then metolachlor had 3 metabolites that were detected at substantially higher levels than the dotted line, which is the parent compound. So these are important to consider for future immunotoxicological studies when we do the wild fish, and also have to consider that these aren't the only chemicals that they're exposed to. They're also exposed to bioaccumulative chemicals such as PFAS and mercury, which we can actually measure in the plasma and their other tissues. So in summary, what makes this work really challenging is also what makes it really important. Our work utilizes field-based immunotox methods with a non-model species. So that's important because there are so many species-specific differences in immunology that comparing what's already in the literature to a smallmouth bass, it just doesn't work. Our study also considers many key metrics, so the sex, the age, season, everything I showed you in that picture, employing indicators like the Health Assessment Index, also analyzes environmental stressors like contaminant exposure, and it features a unique long-term monitoring program which distinguishes it from other studies in the field, integrates multiple biological levels, like Heather was saying at the beginning, the macroscopic, microscopic, cellular function, gene expression, to look at those sublethal impacts and adverse effects that may come from putting it all together. And then also the laboratory-based component that we have. So in the future, I'm still working on the laboratory component and the in vitro exposures. And also for for the field portion of it, going back to those simple correlations and putting them in more advanced statistical analyses to bring it all together. Also developing methods to look at epigenetic analyses and also non-lethal sampling, which Lindsay is going to discuss for her project at the end. And then I'm also working on the pathways analysis for these gene transcripts. So the gene transcripts that we have, they haven't been annotated, so it would be really important for us to be able to put these together and see if there are specific pathways that are being affected more than others or being impacted more than others. And that is the end of my presentation. Thank you.
Great, thank you. I'm always impressed with the approach your team takes to sorting out these complicated ecological issues. Folks, we have time for one really quick question if somebody would like to unmute themselves.
I have a question. This is Irene Salinas.
Hi.
Great talk. Thank you so much. Sorry, I will put my video on as well. I had a question about, I may have missed it, your ROS assays. Are you guys doing that in head kidney leukocytes or in what organ?
Oh, I didn't say, but yes, we're doing a head kidney leukocytes.
Okay. And did you guys look at the proportions of myeloid cells versus lymphoid cells in those? Like you can have higher or lower ROS measurement just based on the proportions of the cells that you have in the head kidney rather than the activity within each cell, right?
Right. I haven't separated that out for just because the method is to separate the granulocytes, and also when I gate it on the flow cytometer, the imaging flow cytometer, I can actually gate out the lymphocytes. So
Nice.
I try to keep them separated since they're part of the adaptive immune response.
Yeah, so you don't know if you had just more myeloid cells anyway or less.
Yeah, I'm not sure.
Yeah, that'll be another thing that I'm curious about is whether or not maybe the chemicals, I guess you'll see that in the in vitro assays, you may just have like some toxic effects on the leukocytes and you have some cell death based, just because you're exposing to the chemicals. And that if you have cell death or apoptotic bodies in the head kidney, you'll have phagocytosis of the apoptotic bodies anyway, because of the clearance of the debris, which will result in higher ROS rather than infection or whatever else. But yeah, that's really interesting.
And that is one benefit of the imaging flow cytometry is because I can take out the apoptotic cells and I don't have to consider them in the gating. Because I can actually see the bright field images of the cells and I can use the machine learning to give me the cells that are together. If that makes sense.
Yeah. Thank you so much.
Thank you.
All right, great presentation. And folks, if you've got other questions, there was one that came in just to me. I dropped it in the chat, and perhaps Cheyenne can answer those there. So virtual round of applause to you.